Dear colleagues, I'm glad to take part in the first electronic international conference on entomology, and I thank the organizers for inviting to deliver the talk. I'm going to talk in advance and pre-record my speech, as during the conference days I will be in a remote corner of Russia doing my fieldwork with hardly any internet access. So I'm going to present the study entitled The Invasive Horseshoesnot Lithmanin Cameraria ridella, Lepidoptera gracilarida, in the southern Russia, the preliminary molecular genetic characterization. This is an ongoing study that has been done by the group of co-authors presented on this slide. I am Natalia Kirichenko, the presenter from Sukachev Institute of Forest, Russian Academy of Science. My co-authors are Natalia Karpun and Lydia Samarina from Federal Research Center, the Subtropical Scientific Center of the Russian Academy of Science from Sochi, Evgeny Akulov from All Russian Plant Quarantine Center, the Krasnoyarsk branch, and Nikita Mamayev and Dmitry Mussolin from St. Petersburg State Forest Technical University. I am going to talk about an invasive species, and first of all I would like to give some words about the modern problem regarding the biological invasions. So I guess many of you know that biological invasions, these are the major component of global change, and what the impact can invasive species have. In fact, they can displace native species, they can reduce native wildlife habitat, we can reduce forest health and productivity, as well as alter ecosystem processes and degrade recreation areas, etc. There are a number of factors that can facilitate introduction, establishment, and acclimatization of insect pests. In particular, these are different human activities, growing international trade, growing tourism, as well as ongoing climate change. In Europe, it has been analyzed the number of records of alien arthropods in the last several centuries, in fact since the 16th century, and the exponential trend was detected. And the similar situation was recorded for the southern Russia. In fact, the number of alien phytophagous insect pests increased significantly since 1816. Since then, the number of records has been known. And the number of records of invasive organisms continue increasing, especially in subtropical area of southern Russia and especially in the Black Sea coast of Russia, as shown on the graph, which was taken from Karpun 2018. In fact, the southern part of Russia serves a particular getaway region for many invasive alien species. Due to high tourism in this area, the southern Russia is well-known resort area, and many Russians tend to go there for spend their holidays and vacations. And a number of invasive organisms arrive in this area with tourism, with different transportations, as well as with plans for planting, taking into account that this region has been very much modernized during the last 2 decades, especially the region of Sochi, preparing for the Olympic Games. Alien insects can rapidly invade huge areas from their relatively small native range, and they can also be very successful colonizing new host plants and become pests in the new environment. Here the scheme is shown regarding the invasion of insects. So in native range, they can be a secondary pest or can be a rare species, whereas during the invasions in the invaded regions, they can multiply their populations in case they are acclimatized in the region successfully, and they can provide a significant damage in the in the secondary range, in the invaded ecosystems. So leaf-mining insect pests, these are particular group of organisms. This is an ecological group of arthropods where damage leaves. We are endophagous species because their larvae live inside the leaf tissue, making so-called mines, cavities inside the leaves. Here the leaf mine is shown. If cut across, you may see the mine cavity, which is protected by the epidermis of the leaf. What is the special about leaf-mining insects? In fact, this is a very big group of insects, and there are a number of species from different orders of insects which have a leaf-mining lifestyle. For example, some Lepidoptera, moths, some Coleoptera beetles, as well as some flies and sawflies. So this group is very rich in diversity. Number of species have a vast distribution. There are a number of ornamental and agricultural pests known among leaf miners, as well as some insects in this group can be highly invasive. In particular, leaf mining moths are of very special interest. These are Gracillariidae moths. There are a number of invasive leaf mining moths which penetrated southern Russia, and they are listed on the slide on the right, on the left. And among them, I will briefly introduce the insects which are widely known in Russia due to their outbreaks in the present time. For example, the lime leaf miner Phalanorictor issiki, which originates from East Asia. In the last 2-3 decades, it invaded nearly the whole Europe, Western Russia, and Western Siberia, where it attacked native limes. And nowadays, it's known as an ornamental pest. Among others, this citrus leaf miner, Phylacnistis citrella, which originates from Southeast Asia. And presently, it is known around the whole world where citrus is grown. And in fact, this leaf miner is a serious agricultural pest on citruses. And our target species is a horse-chestnut leaf miner, Cameraria oridella. It is presently widely known across all Europe and as well as in western part of Russia. It attacks horse chestnut, Aesculus hippocastanum, and this insect is known as an ornamental pest, providing spectacular outbreaks. In fact, this species was detected in Europe, in Macedonia, near Orchid Lake in 1980s, and notably already in a high density. Formally, it was described in 1986 by Deshka academic. Presently, the horse chestnut leaf miner is found nearly in all European countries. Here, the schematic map is provided showing the distribution of the pest in the last 25 years across Europe. And this insect also invaded Russia. For the first time, it was detected in 2003 in Kaliningrad, the city of the Kaliningrad region, which is situated in Eastern Europe. In 2 years, in 2005, it was already detected in Moscow, which is about 900 kilometers far from Kaliningrad by starlight. And in the following years, the insect distributed across most part of the European part of Russia. In 2014, it was detected in the southern Russia, in particularly in the region of Sochi. In 2018, rather recently, it was documented in the lower Volga region. And I can say that is more or less penetrated all the corners in a European part of Russia where its host plant is grown. And for this insect, a very interesting study was done in Europe, aiming to detect the genetic diversity of the pest. So the molecular genetic study, which was run in the modern range of Horseshoesnot Lithmanin, revealed high genetic diversity in the Balkans, suggesting that the Balkans can be the moth's motherland. On the contrary, in the other parts of Europe, where the species is known as non-native, it has a low genetic diversity. So this data is taken from Valade et al. And other studies, which were done a few years later using the historical herbaria, also supported the hypothesis about the Balkan origin of this pest. So the aim of our study is to characterize Cameraria oridella's populations genetically in order to identify the invasive haplotypes in Russia. So here we present the preliminary results of this research because this study, as I told at the beginning, is an ongoing study. So the Horseshoesnot Lithmanin was sampled in Russia, in particular in the southern regions, in the Republic of Adygea and the Republic of Crimea, and in central Russia, in particular in Moscow. So about by 20 larvae or pupae from mines were dissected on a Horseshoesnot leaves, and these specimens were preserved in 96% ethanol solution for following molecular genetic study. In addition, we also sampled damaged leaves, and we placed them in annotated herbarium collection in case we need additional specimens. We can dissect them from dried leaves. So the specimens which were collected were subjected to the DNA barcoding. So we sequenced CO1 of the mitochondrial DNA. And the sequencing was done at the Canadian Center for DNA Barcoding, at the Biodiversity Institute of Ontario, at the University of Guelph in Canada. So the identification of the specimens was done by the DNA barcodes in bold. The barcode index number was retrieved for the species. Additionally, we borrowed 4 CO1 sequences of Cameraria oridella from Europe, publicly available in bold. In particular, we used the 2 sequences from Belarus, from Minsk, sampled in 2006 by Lipinskaya, one from Macedonia, Kisevo, sampled in 1985 by Deskar, and 2 from Greece, Jesale, collected in 2008 by Lopez Wamonte and Augustin. So Macedonia and Greece, together with other Balkan countries, are considered to be the native range of the species. So the obtained sequences were processed. For sequence alignment, we use bioedit. The phylogenetic tree was constructed in MegaX using the Kimura 2-parameter model and displayed as a maximum likelihood tree with 2000 iterations and a bootstrap support. But the intraspecific genetic distance was estimated using the same models as said above. We identified haplotypes by comparing what we obtained with the haplotype set identified for the species in the study published by Valade and Kotas. And for routing, we used the sequences of the 2 specimens of East Asian Cameraria nipponica, which was collected on Acer caudatum and Acer pseudozeboldianum in the Russian Far East by myself in 2016. So here we present the fragment of the tree we obtained by analyzing the sequencing data of the Cameraria oridella from Russia. So on the fragment of tree represented on the slide, by blue you can see the specimens which originate from Russia, and the black specimens, these are those were retrieved from Bolt, and we use them as a reference sequences. As you can see, the sequences from Russia and from Europe, they all made a one cluster which corresponded to Cameraria oridella, and in Bolt database it was assigned to one bin of the species. So, and we can say that the analyzed DNA barcoding fragments, we did not show any divergence in those analyzed Russian specimens represented on the tree fragment, and furthermore, they were identical to that of the European specimens involved in the study, and all of them represented one haplotype A, which is most commonly found in the modern range of the species in Europe. So our preliminary study based only on 4 individuals shown in this presentation, randomly sampled in the 3 geographical distant regions, the distance between the regions about 400 kilometers in a straight line between Crimea and Adygea, and more than 1,000 kilometers between Moscow and Crimea and Moscow and Adygea. So far revealed only one, the most invasive haplotype. We believe that the further sampling and detailed analysis will highly likely return similar phylogeographic pattern as it was identified in the neocolonized regions of Europe. And according to Valade and co-authors, the detected haplotype A was the dominant in the Balkans and the invaded regions across Europe. However, in the Balkans, another 25 geographically structured haplotypes were revealed in the MOVS population. High haplotype diversity in the Balkans and the low haplotype diversity in other parts of Europe signal about rapid expansion of Cameraria oridella across European continent. We believe that it is also the case for the European part of Russia, where the pest colonized the significant area in the less than 2 decades. And I also would like to underline that the Horseshoesnot Lithmanin is getting an outbreaking species, especially in the southern Russia, where it provides spectacular outbreaks. In the 7 years after its first detection in Sochi in 2014, Cameraria oridella became a major pest over Horseshoesnot, and in 2014-2015, less than 1/4 of the leaves carried mines in the experimental parks in Sochi. But in the following years, we observed the sharp growth of the pest population, and by 2020, the damage was so much severe in the biggest part of the city, with more than 75% of leaves carrying numerous mines, affecting the appearance of the trees in the parks, and somehow badly affecting the view of the trees in the urbanistic area. So for the conclusion, our preliminary molecular genetic study based on the analysis of C1 mitochondrial DNA gene in a few individuals sampled in Russia, in Moscow, the Republic of Crimea and Adygea, indicates the presence of one haplotype largely found across the secondary range of the species in Europe. The vectors facilitating the pest distribution in Russia, as well as the moth's ability to establish the novel trophic associations with exotic Sapindaceae plants, to which Horseshoesnot also possess, extensively cultivated in the botanical gardens and the parks in the southern Russia, in particularly East Asian maples, remain unexplored. The study we are presently running in the southern Russia will help to clarify this and other questions about the Cameraria oridella invasion in the country. So we would like to thank the colleagues who helped us with sampling of Cameraria oridella in Moscow and the Republic of Adygea, and we are also grateful to the team of the Biodiversity Institute of Ontario at the University of Guelph in Canada for their great assistance in the production of DNA barcodes. This study was supported by the Russian Science Foundation. Thank you very much for your attention. Unfortunately, I won't be able to answer your questions immediately. So in case you have further questions or you'd like to discuss, you may please contact us by emails listed on this slide. Thank you very much.